Okay, welcome everybody. In the interest of time, we're going to get started. So welcome, my name is Victoria McPhail. I'm the coordinator of the Center for Bee Ecology, Evolution and Conservation. So I want to welcome you to our kind of internal BEEc Associates Speaker Series. This is held every month at 11 o'clock on the second or third Friday of the month. It kind of varies a little bit. So we have today on February 17th, we have Tom, and then on March, we have Bahir, and then following up in April, Matilda. So follow your emails. All the BEEc associates get the email with the Zoom link each month, but it's the same link each month as well. So when you just put in your calendar, you'll have that going forwards. Additionally, for the associates, we do send a newsletter out by email, but you can always find past issues on our internal website. That's yorku.ca/b/members. There's information there about the newsletters, our speaker series, links to some of the past presentations, as well as those are found on our YouTube club, our YouTube channel. And if there's any interest in individuals having like a book club or a journal club, let us know and we can start getting that set up. I'll remind you that as per most webinars, please mute your microphone during the presentation and save your questions until the end during our Q&A session. Tom has given us permission to record this meeting and will be posted to YouTube later on. So please keep that in mind if you want to share your screen or do a video audio sharing later on. You can always use a chat box as well to share your questions at the end. I do also want to acknowledge the land that we are on. So we acknowledge that many Indigenous nations have long-standing relationships with the territories upon which York University campuses are located that precede the establishment of York University. York University acknowledges its presence on the traditional territory of many Indigenous nations. The area known as Tikaranto has been taken by the Anishinaabeg Nation, the Haudenosaunee Confederacy, the Huron-Wendat, and the Métis peoples. It is now home to many Indigenous peoples, but we acknowledge the current treaty holders, the Mississaugas of the Credit First Nation, and note that this territory is subject to the Dishes One Spoon Wampum Belt Covenant, an agreement to peacefully share and care for the Great Lakes region. I encourage you to check out www.native-land.ca. Learn more about the peoples who are currently on the land that you are researching or you are currently located on, as well as the past traditional lands that people may have had in your area. So I want to bring it to today's talk on the effects of dune stabilization on bees and aculeate wasps in the southern Canadian prairies. So Tom Onuferko developed a professional interest in bee diversity while working towards a Master of Science degree under the supervision of Professor Miriam Richards at Brock University in St. Catharines, Ontario. He subsequently joined Professor Lawrence Packer's lab at York University in Toronto, in pursuit of a PhD focusing on the systematics and evolutionary history of the kleptoparasitic bee genus Aculeates. He then proceeded to document and study the aculeate fauna and disappearing sand dunes across the southern Canadian prairies as a postdoctoral Research Fellow for Species Diversity, this species discovery at the Canadian Museum of Nature, and this project will be the focus of his talk today. He's currently a research associate with the museum and has recently joined Professor Scott McIver's lab, the University of Toronto Scarborough, as a CERA Postdoctoral Research Fellow to study changes in wild bee diversity in response to various environmental stressors in Southern Ontario. Thank you very much, Tom, and welcome, and take it away.
Just unmute myself first, and I'm going to share my screen. It's this one. Let me see if that's the correct one.
Yeah, looks good. Just have it in slideshow mode.
Yeah, okay. Is that okay? Everyone can see?
Yeah, it looks good to us.
Slides. Okay, well, hello. It's a pleasure for me to talk to you today about the work I did as a BD Postdoctoral Research Fellow at the Canadian Museum of Nature on the effects of dune stabilization on bees and aculeate wasps in the southern Canadian prairies. So I'm just going to minimize my own camera because it's blocking me from seeing my own slides. Okay. So I would like to begin by acknowledging that the land on which this research was carried out is the traditional territory of many Indigenous peoples including Anishinaabe, Assiniboine, Cree, Métis, Niitsitapi, Nakota, Saulteaux, and Tsuut'ina peoples. To give you a bit of background, in Canada's prairie provinces, there are unique tracts of inland sand dunes commonly known as the prairie sandhills. These trace their origins to the Quaternary glacial apex when grinding ice, namely the large Laurentide ice sheet, and weathering reduced bedrock to gravel, sand, and silt. Unlike coastal and desert dunes, the prairie sandhills are in otherwise largely vegetated inland areas, and they're comprised of a comparatively limited supply of sand. And they rely on a certain level of wind erosion to generate loose sand to be reshaped into dunes, which often take on a characteristic parabolic shape, as exhibited by this large isolated sand dune at Douglas Provincial Park in Saskatchewan. Extensive dune fields are found across North America's Great Plains, as you can see from the black areas in this figure, published in a paper by Hugenholtz and colleagues. And these were potentially active at various times over the past 13 or more millennia. Today, active sand dunes in the prairie provinces are mostly located within the prairie grassland and parkland natural regions, which are indicated by the lighter shades of gray in this figure. And these dunes are patchily distributed. In fact, when you visit some of these places, it almost looks as if someone just dumped large piles of sand in the middle of a grassland. It's really quite extraordinary. And so, due to their isolation, the prairie sandhills are islands of biodiversity. They're threatened by stabilization, which is the fixation of sand in place and eventual conversion to grassland by succession by erosion inhibiting vegetation. They are also home to a wide range of sand-loving taxa called samophiles, including dune-restricted species that have been assessed as rare, threatened, or endangered within Canada. Well-known examples include specialist neptuid moths of the genera Copoblepharon and Skinia, the latter of which is illustrated here. This is the gold-edged gem, which is currently listed as endangered within Canada. Now, the reason these and other dune-adapted taxa are of conservation concern is because over the past century, sand dune activity has greatly declined in the southern Canadian prairies, particularly in southeastern Alberta and southwestern Manitoba, in areas referred to as the Middle Sandhills and Brandon Sandhills, respectively. And the widespread vegetative stabilization of dunes in recent years has been attributed to a multitude of causes, and these may be categorized as non-climatic or climatic, with the former being much more consequential. The main non-climatic causes, which are a consequence of European settlement, include the use of extensive irrigation and suppression of fires to reduce soil losses by wind erosion. Apparently, this was in response to the Dust Bowl of the 1930s. But also the disappearance of grazing migratory bison herds, which were some of the major destabilizers of active sand dunes. And so the natural disturbances that inhibited plant growth in these areas in the past are no longer present. The areas in which the remaining dunes occur are now largely used as rangeland for cattle grazing and natural gas extraction, but some have been set aside as conservation areas and or military bases. Visitors also use the prairie sandhills and surrounding grasslands for recreational activities, including ecotourism, but also hunting, which isn't always legal. The non-developed areas surrounding the dunes comprise diverse habitats that are also quite rich in wildlife. These are just some of the interesting animals I encountered in the grasslands surrounding dunes I visited in Alberta and Saskatchewan. I didn't want to take a closer picture of the rattlesnake for obvious reasons, so that one came out a bit blurry. By the way, these two species are listed as special concern in Canada's Species at Risk Public Registry due to the disappearance of much of their original grassland habitat. Although home to a wide range of sand-adapted taxa, terrestrial arthropods account for most of the animal diversity in prairie sandhill environments. Despite their diversity, sandhill arthropods aren't very well studied, except for some of the more charismatic groups like tiger beetles. And the dunes are an especially rich habitat for solitary bees and hunting wasps, which together with ants comprise the hymenopteran infraorder Aculeata. And open sand patches have several uses for aculeates. They are used by species like the sand wasp Bembex pallidopicta to search and compete for mates. Sand, of course, serves as a nesting substrate for various bees and wasps. And finally, they serve as sources of food. Wasps hunt or scavenge for other arthropods on sand or samophyllous plants, including, for example, this Ammophila with her captured caterpillar prey. And both bees and wasps actively visit flowers of plants growing on or at the edges of active sand dunes, including, for example, these prairie clovers, which almost act like a magnet for pollinating insects. Despite the significance of sand to aculeates, there is limited information on the effects of declining sand dune activity in the northern Great Plains on the structure of aculeate communities. The most important assessment published so far is a report by Albert Finnemore and Don Buckle that documented wasp diversity in the middle sandhills of Alberta. Therein, the authors reported that wasp richness was higher in sites with sandy soils as opposed to black soils, or chernozem as they referred to it, that stabilized dunes had more wasp species than active dunes, and that species turnover was very high between active and stabilized dunes. But this study did not consider the bee fauna, unfortunately, and it had a limited geographic scope that was focused on the Canadian Forces-based Suffield National Wildlife Area. So in a cross-sectional study, I surveyed both bees and aculeate wasps at 13 dunes at various stages of stabilization on a larger scale from southeastern Alberta to southwestern Manitoba. Every site was visited 5 times in the spring and summer of 2019 for a total of 65 collection events. That meant a lot of driving and hiking that year, but my carbon footprint was mitigated somewhat by my being much more sedentary during the following years of the pandemic. Sampling consisted of a combination of pen trapping and time-targeted netting. The former involved setting out 30 pan traps at each site along an X-shaped transect for 6 to 7 hours per collection event. This transect pattern was chosen to maximize coverage of a dune. And the latter served as a supplemental method to detect species that might be missed by the former. As we all know, pan traps bias collection towards certain species and don't capture others very well. And so essentially all aculeates were surveyed with the exception of ants. And this is because the small size and terrestrial and fossorial nature of ants made it impractical to sample them in targeted collections, and also because their numbers in pan traps were strongly influenced by the proximity of individual traps to ant nests. To study the effects of stabilization, 2 measures of the variable were considered, one qualitative and one quantitative. The qualitative measure was stage of succession or stabilization level, which was visually assessed based on the availability of areas of open sand, regardless of vegetative cover. And most of the sample dunes were active. They were recognized as such by the presence of large areas of open sand. 3 were identified as stabilized with no open patches of loose sand. Here you can see an example of a stabilized dune in Saskatchewan's Great Sandhills. Although you can clearly see that the soil is sandy, it's quite hard when you walk on it compared with the loose sand found on active dunes. And then another 2 dunes were regarded as stabilizing, and by that I mean intermediate between active and stabilized, with open patches of loose sand in otherwise largely vegetated areas. And the quantitative measure that was considered was percent plant cover. And this was estimated at each site in 5 1-meter by 1-meter quadrats on the last day of sampling. This was in August when vegetative cover was at a maximum. Floral composition was also considered. Rather than struggle with plant identifications on site, which would have been far too time consuming and not a very efficient use of my time, I simply imaged the quadrats with my phone and collaborated with a botanist at the Canadian Museum of Nature, Paul Sokolov, who identified the image plants to the lowest taxonomic level possible. And then together we estimated percent plant cover to the nearest 5% in each quadrat, from which we produced average estimates of vegetative cover for each site. Overall, more than 12,000 non-ant aculeates were collected, of which about 63% were bees and 37% were wasps. The collection, which is now more or less properly curated, fills nearly 2 large insect cabinets, each holding 20 drawers. Although fewer in number somewhat, wasps are more diverse than bees in terms of family and generic richness, but the 374 species sampled were evenly divided between the two groups. This figure shows differences in the numbers of specimens and species sampled using both active and passive methods, with yields presented separately for each pan trap color, blue, white, and yellow. As you can see, white pan traps were the most effective within these sites. And although I have comparatively little to show in terms of absolute numbers for the nearly 50 hours spent on targeted collections, a lot of species were still collected this way, including 19 that were missed in pan traps. Notable and unexpected findings include one new species of sweat bee, which is known only from active dune systems in southern Alberta and southern Saskatchewan. It was identified as a new species by Joel Gardner and Jason Gibbs, who examined the material I collected and described the species in 2021. And I'm very honored that they chose to name it after me. Represented by 360 specimens, Laesioglossum onuferkoi was one of the 5 most abundant bees I collected. Otherwise, the species is represented by just 17 known specimens in existing museum collections. And those were collected by Albert Finnemore and Darren Pollock in the 90s in the Canadian Forces Base near Suffield, Alberta. According to one of my collaborators on the study, Matthias Buck at the Royal Alberta Museum, there are probably 3 undescribed species among the wasps I collected of the genus Diodontus. And it wouldn't surprise me if there were undescribed species among some of the larger and taxonomically problematic bee and wasp genera that were sampled, such as the bee genera Andrena, Nomata, and Osmia. In addition to the new species, many were previously unreported from the provinces of Alberta, Saskatchewan, and Manitoba, at least at their time of capture. Overall, a whopping 183 new provincial records and 21 new records for Canada were identified, although some of these are also known from separate unpublished surveys. This protocephalic specimen is an example of a new record from Manitoba and was included in the checklist of the bees of Manitoba published by Gibbs et al. Just this month. Now, it is obvious that many of these records simply reflect a lack of extensive surveying across a larger region historically, especially in Saskatchewan, where most of the new records are from, as well as a lack of available expertise to identify previously vouchered specimens. But still, it is notable that 22 of the WASP records appear to be unique to my surveys, as my co-author Matthias Buck has not come across those species in exhaustive searches through existing museum collections across Canada. Fourteen of the sampled species were identified as open sand specialists based on the literature and/or independent observations of these species occurring exclusively on dunes, blowouts, and large open patches of loose sand, at least in Canada. Most of these are apoid wasps, but some bees and vespoid wasps, and I use the term vespoids in the broad sense here, have also been identified as open sand specialists. And so these 14 species comprised nearly 1/5 of all sampled specimens, but less than 4% of the number of species detected. Looking at floral composition, at least 18 different plant species were present in the sampling quadrats. Although some species will have been missed by the quadrats, the more abundant ones were undoubtedly detected. The dominant species was Ladenia lanceolata, a legume commonly known as lemon or dune scurf pea. I will be using the name dune scurf pea throughout, as this fits much better with the theme of this particular study. And this species was detected at 11 of the 13 sampled dunes and accounted for about 43% of the estimated vegetative cover across the 65 quadrat samples taken. This is an early successional species on inland sand dunes that typically dominates the pioneer stage, but is replaced by other plants in subsequent stages of succession. Grasses, which were mostly represented by Canada wild rye, Indian ricegrass, prairie sandreed, and wheatgrass, accounted for most of the remaining estimated percent vegetative cover at about 44%. The stabilized dunes were all dominated by grasses, whereas dune scurf pea was often dominant in sites with open sand, although not always. And one dune dominated by dune scurf pea had almost 80% estimated vegetative cover. Using rarefaction to adjust for differences in sampling effort, we can see that aculeate species richness are more or less the same among the different stabilization categories. Singletons, which were represented by 106 species and morphospecies, accounted for less than 1% of the sampled specimens, but more than a quarter, 28.3% to be precise, of the species detected. Now, analyzing the bee and wasp data separately, we can see that the ratio of bee species to individuals was highest in active dunes, whereas the ratio of wasp species to individuals was highest in stabilized dunes. And the latter pattern is particularly interesting because it's consistent with, or at least does not contradict, the findings of Finnemore and Buckle, who reported that stabilized dunes in Alberta had more wasp species than active dunes. Comparing the abundances of bees and wasps, here we see that the proportion of wasps decreased relative to bees with stabilization from nearly 40% to less than 30%. And not surprisingly, we also found that the relative abundance of the 14 species we identified as open sand specialists decreased with stabilization. These comprised almost a quarter of the specimens sampled on active dunes, but less than 7% of those on stabilized dunes. Now you might be wondering, why would there be any open sand specialists on stabilized dunes? Those presumably are transient individuals, given that stabilized dunes were adjacent to or within 100 meters of active dunes. And wild bees and aculeate wasps, of course, are known to range to forage at much greater distances from their nesting sites and disperse over fairly large distances. Comparing the abundances of bees and wasps in sites dominated by dune scurf pea and versus sites dominated by grasses, we see that the proportion of wasps relative to bees was somewhat lower in sites dominated by grasses. And here you can see that the proportion of individuals of species identified as open sand specialists was also lower in sites dominated by grasses. I should note that although dune scurf pea was found to be dominant only on active and stabilizing dunes, grasses dominated in sites assigned to all three stabilization categories, so this variable isn't merely another indicator of stabilization, or at least not a very reliable one. The effects of dune stabilization level, dominant plant type, and other variables and interactions thereof on these different subsets of the data I just showed you were formally tested using non-parametric permutational analyses of variance, Permanova, each with 10,000 permutations. In each analysis, a Bray Curtis dissimilarity matrix of bee and/or aculeate wasp species composition was computed from species abundance data. And in all analyses, permutations were constrained within sites. Here we can see variation in the composition of bees, wasps, open sand specialists, and non-specialists among sand dunes at three different stages of stabilization in the top row, and sites dominated by dune scurf pea and sites dominated by grasses in the bottom row, visualized through two-dimensional principal coordinates analysis. And in all subsets of the data, the main and interaction effects of dune stabilization level and dominant plant type were highly significant. Now, to understand the relationship between percent vegetative cover and abundances of bees and wasps, a generalized additive model was tested, treating percent plant cover as a smoothing term. Smooths were produced separately for each combination of aculeate group, ecological group, and dominant plant type. And what this figure shows is that wasp abundances varied non-linearly with percent plant cover in sites dominated by grasses, with numbers peaking around 50% estimated vegetative cover. And the overall abundance of wasps identified as open sand specialists decreased with increased percent plant cover in sites where dune scurf bee was identified as the dominant plant. And no significant relationship between overall abundance and percent plant cover was detected for any of the other combinations of these three variables: aculeate group, ecological group, and dominant plant type. Looking at the main effects included in this model, we see no differences in the overall abundances of bees and wasps, but that there were far more individuals of non-specialist species than species of open sand specialists on the sampled dunes, which we already knew. We also see that there were more aculeates on average in the 6 sites where dune scurf bee was identified as the dominant species than in the 7 sites dominated by grasses. Generalized additive models of species richness were also tested, treating percent plant cover as a smoothing term. Neither bee nor wasp richness varied significantly with increased vegetative cover when all sites were considered. As shown here on the left, these are the results from Model A. Model B, which produced smooths separately for sites dominated by dune scurf bee and sites dominated by grasses, was a much better fit for the data, and showed that species richness varied non-linearly with percent plant cover in sites dominated by grasses, and the observed relationship resembles those between the overall abundances of sand specialist and non-specialist wasps and percent plant cover in those same sites, with numbers also peaking around 50% estimated vegetative cover. In terms of beta diversity, taxonomic turnover was generally lowest between the sites that are closest to one another, regardless of stabilization level. The 5 pairs of sites most similar to and within just a few kilometers of one another are shown here by the blue brackets. And in only one case are both sites in the same dune stabilization category. I am referring to the 2 active dunes near the town of Webb in Saskatchewan at the top of this dendrogram. And this pattern contradicts the findings of Finnemore and Buckle, who reported lower turnover among stabilized dunes and among active blobs, and a higher turnover between active and stabilized dunes. And this inconsistency can be explained by the much larger spatial scale at which dunes were surveyed in the present study, with bees and wasps drawn from markedly different species pools between the widely separated areas. For reference, whereas the shortest distance between any 2 sampled sites was a little less than a kilometer, the sites at Spruce Woods Provincial Park, listed at the bottom of this dendrogram, the maximum was almost 800 kilometers. And whereas the greatest distance separating any paired sites, and by that I mean sites that were sampled concurrently on the same days, even though that was just a little over 3 kilometers, the shortest distance separating any sites that were sampled on different days was about 18 kilometers. So all that is to say is that had this been a local as opposed to regional study, with all sites in fairly close proximity to one another, then I'd expect for species assemblages to have been much more similar between dunes at similar stages of stabilization. And so to summarize, it is quite clear that the effects of dune stabilization are not the same for different bee and wasp guilds, and that stabilization changes the structure of dune inhabiting aculeate communities. As expected, Psammophilus species and I use the term Sandhills in a very restricted sense here, decreased with dune stabilization. And although the 14 species of open sand specialists are potentially at risk from extirpation in areas where dune activity continues to decline, most are widely distributed in North America, and none have been assessed as threatened or endangered within Canada. And so with that in mind, these species have the potential to serve as a monitoring tool for changing habitat quality due to their co-occurrence with at-risk taxa like dune-dependent moths and tiger beetles, which for obvious reasons should not be lethally sampled. Percent plant cover alone was not found to be a reliable indicator of aculeate abundance or richness across guilds or dune systems. As we saw, bees and aculeate wasps responded non-linearly to percent plant cover on dunes dominated by grasses, but not dune scurf bee. And so the extent to which the dominant plant or plants cover a particular dune seems to better explain the numbers of individuals and types of aculeate species it can sustain. We also saw that abundance and richness were generally high on dunes where the dominant plant was dune scurf bee. Now, the extent to which the plant itself is responsible for the observed higher levels of aculeate abundance on dunes where it appears to dominate is unclear. Dune scurf bee flowers are visited by bees, and insects associated with the plant may be an important source of prey for aculeate wasps, but dune scurf bee may just be an indicator of high quality habitat for bees and wasps, just because it happens to grow where there are abundant food and nesting resources. And while sand dunes in this region remain under threat, long-term effects on samophiles and especially dune-dependent species should, of course, be monitored. And to that effect, our study provides a preliminary checklist of bee and aculeate wasp species in prairie sandhill environments, which I hope will be used as a baseline for future surveys. And if there's one final message with which I'd like to leave you, it's that these are really spectacularly beautiful places. I very much encourage others to visit and study them. And of course, that we ought to do our best to preserve the prairie sandhills as they comprise a truly unique and remarkable habitat within Canada. And so with that, I would like to thank the following sources of support. My former collaborators on the study, Dr. Matthias Buck at the Royal Alberta Museum, Professor Jason Gibbs at the University of Manitoba, and Paul Sokolov at the Canadian Museum of Nature. I sincerely thank the Ross Beatty family who funded this fellowship for the purpose of supporting research that addresses conservation issues within Canada, and the Canadian Museum of Nature through which the fellowship was offered. I thank several individuals for assistance with lab work and logistical support, including Dr. Bob Anderson, who was my supervisor. I also thank Roger Bull and Anna Ginter from the CMN Laboratory of Molecular Biodiversity for assistance with the DNA barcoding that went on there, which I used as an identification tool for a subset of specimens that were difficult to identify morphologically. I also thank Dr. Rob Longair for graciously volunteering his time to help label and do some preliminary sorting of specimens. The lands on which some of the dunes were, the areas in which some of the dunes were included provincial parks, and I thank the provincial governments of Saskatchewan and Manitoba for granting me permission to sample there. I also learned before conducting field work that one cannot simply go and lethally sample insects anywhere within the province of Saskatchewan without an academic research permit from the province, including on private lands. And so I'm grateful to the provincial government of Saskatchewan for allowing me to sample insects lethally. And many of the dunes were on privately owned or leased lands. And I thank several individuals for granting me access to those dunes, as well as to the roads that get you as close to the dunes as possible, which saved me a lot of hiking time. And I thank everyone who attended the seminar for their interest in my research, and the BC group at York for hosting me and organizing the speaker series. Thank you, and I'm happy to answer any questions at this time.
I'll just remind people that we do have next talk in the series. I'm trying to find my slide here. Is next month on the 17th by Dr. Bahir Bhattar, the evolution of protein soup in male ejaculate. So getting into sexy talk, seminal fluid proteins, learn all about them next month at 11 a.m. And then the month after that, we have Matilda Tissier, April 14th. I don't have her talk title yet, but I'm sure it'll be really interesting. So just stay tuned to your email for that Zoom link. It's the exact same link each month. So if you have it for today's talk, just put in your calendar with the date and join in next month. And with that, I thank you all for joining. Thank you for your questions and discussions, and we'll see you all next month.
Thanks all.